Dear students, hello, hello and good afternoon. Please settle down quickly. I can see that you are eating your lunch, but I think your break's over now. So let's settle down quickly and let's get started with the topic. Alright, so yeah, okay. Centers, please report your attendance and we are beginning right away. So, dear students, in the last class we had covered up till the long distance. We had actually started the long distance transport of water, transport in plants. Alright. So, when we talk about transport in plants, I had told you that short distance transport had already been covered, right? Osmosis, water potential, used to measure osmosis. Then we talked about diffusion, facilitated diffusion, and also the active transport. All these topics have been covered, so short distance transport has basically been covered. Now what remains is long distance transport of substances, and long distance transport includes transport of water through xylem and transport of food substances through phloem. Alright. So in the last class yesterday I told you that there were three important things in a transport theory of water, and that is capillarity force, that is cohesive force, that is adhesive force. All these three forces together help in making a continuous water column from the root till the leaf surface, right? And it forms a sort of unbreakable capillary, continuous capillary, because as the water is evaporated, it does not break, it absorbs fresh water molecules from the soil surface, and here is how this is how the whole process takes place. Okay, so I gave you a good explanation of all these factors, and I also told you that there is another thing called root pressure theory. Root pressure is a sort of weak pressure developed by osmosis. It is called osmotic pressure. Root pressure is equivalent to osmotic pressure. Because of this pressure, some substances in some weak herbaceous plants, they develop this tendency to be taken in, and along with the water they come out through specialized water stomata or water excretory stomata. These are called hydrathodes. Okay, and the water looks like minute droplets condensing on the leaf tips. Right? So guttation is this phenomenon, but guttation is not responsible, root pressure is not responsible for the long distance transport of water. Long distance transport of water is explained by Dixon and Jolly's hypothesis. So in the last class yesterday, hello everyone, hello Tamenglong. In the last class yesterday, so we had done this part. So carrying forward, let us do some things. just a second. Yeah. Okay, so I had told you that we had done some things, and all right. So transpiration of water, the transpiration of water, it is basically explained by this root pressure theory, and it says that because of capillarity, because of adhesion, because of cohesion force, the long distance transport of water becomes possible. Okay, long distance transport is possible because of adhesion here. Okay, adhesion, cohesion, and capillarity. Okay, capillarity because the water molecules are sticking to each other like this. They're sticking to each other. They are forming a continuous water column like this. All right. So because of this phenomenon of capillarity, the adhesion plus cohesion plus capillarity equal to continuous water column in xylem, and finally it escapes out from the leaf surface. Evaporation of leaf surface, water from leaf surface, it is called transpiration basically, and this creates a pull called the transpirational pull. So the long distance transport of water is explained by transpirational pull theory by Dixon and Joly. So just revising you what I told you yesterday, transpirational pull theory explains movement of water through xylem. Okay, and this is how we do it. Okay, so some people are still coming in. I would ask you to please settle down quickly. Scientists, I think you can now finally report your attendance, full attendance, because everybody is now present in the class. So do that quickly, please. All right. So transpirational full theory is finished now. Now let us begin with movement of food and other solutes through phloem. So now we have to discuss transport through phloem. Transport through phloem, or long distance transport of food substances through the phloem. Long distance transport of food through phloem. When we talk about phloem, I have already told you in the anatomy of flowering plants, you will have a test of anatomy in flowering plants tomorrow, students, right? Okay, so please be ready for it. So in anatomy of flowering plants, I had told you that phloem consists of 4 cells. It is companion cell, it is sieve tube element, the third cell is phloem parenchyma, and then there is phloem fiber. 4 types of cells are present in the phloem. Out of these, companion cells and sieve tube together, they take part in the transport of the food substances. So this part we have already dealt with in the class. Okay. So let's start how the food substances are basically transferred. Okay. So transport of food substances through phloem, it is explained by the best theory which explains transport of food substances through phloem is Munch's or Cadbury Munch's pressure flow hypothesis. Munch's pressure flow hypothesis. Munch was a scientist. Munch's pressure flow hypothesis. Okay. So do you know about this hypothesis? Munch's pressure flow hypothesis? Yes? Okay. So please write it down first. I'll explain why, what it means actually. Alright, so give me a second. I'm trying to actually give you the idea of alright, so let us see. Okay. And one more thing. All right. So all right. So this is what we have to do right now. The best theory which explains the Munch's hypothesis. So please see this diagram of phloem now. This thing here, this this is a sieve tube element. What you are seeing here, it is a sieve tube element. This is visible to everybody. This is a sieve tube element cross section. I am cutting a cross section through a sieve tube element, and this is how it looks like. So this part, it is actually the sieve plate. The section that is you are seeing here, it is the sieve plate. This is the sieve plate. Can you please scroll up? Yeah, sure. When you are finished, let me know. Okay, all right. So this is a diagram of sieve tube elements and companion cell. So this what you are seeing here, this is one sieve tube element, right? And this part here, it is a sieve plate. Sieve plate has many small pores inside it. In the anatomy chapter, I told you that there is a great difference between the phloem, sieve tube element and sieve cell of gymnosperms. Sieve tube elements are found in angiosperms. In gymnosperms, these are sieve elements only, or sieve tube cells. Sorry, not sieve tube, sieve cells only. What is the difference? Sieve cells of gymnosperm does not contain this sieve plate. Sieve plate is absent there. Now this is a characteristic feature of angiosperms only. And this cell here, I am drawing it with green color. This cell here, it is the companion cell. This is the nucleus. This is the companion cell. Okay. Actually, this is written in French language, cellule compagne. Actually, this is French, but that does not matter to us. Okay. So what we have to do is just concentrate on. Yeah. Now, between the sieve tube cell and the companion cell, there will be plasmodesmata connections. I have drawn them here. Okay, I am drawing some gaps here. I have to just put the eraser. These are plasmodesmata connections, direct cytoplasmic connection between the sieve tube element and the companion cell. Okay, and because of this, both are interlinked to each other. The sieve tube element does not contain a nucleus. Without the nucleus, it cannot survive if it not had been for the companion cell. Companion cell keeps the sieve tube element alive. Companion cell keeps the sieve tube element alive. Alright. So even without a nucleus, it can survive. So you can see this here, that this is the companion cell, this is the companion cell, sorry, this is the companion cell, this is the sieve tube element, and between this is the sieve plate, this is the longitudinal section diagram, this is the transverse section, this is the longitudinal section. Okay, and in this longitudinal section you can see how the sieve plate is arranged. Now this is another transverse section, actual microscopic transverse section of sieve tube cells. Okay, so you can see this is the sieve plate, this is the sieve plate actually. Alright. Now what is going to happen? Let me explain. Please see a short video. It will help you in understanding what is happening. Okay. So I want you to see a short video, and just a second. Okay, so so you can see it with I won't speak here. You just listen to what is being said here. Okay.
Hydrogen ions return to the cell through the protein channels along the concentration gradient. At the same time, sucrose is translocated together with these ions. Sucrose is therefore transported through the membrane of the companion cell to the sieve tube by means of co-transport, which in this case involves simultaneous transport with hydrogen protons. Phloem unloading takes place in the cells of acceptor organs. Like phloem loading, this can take place either via the apoplast or via the symplast. Donor organs that grow most intensively, such as young leaves, are supplied with organic substances by the symplast. Storage organs, however, are supplied by the apoplast. During phloem unloading, sucrose is again translocated by means of co-transport with the help of the proton pump. Parasitic organisms, for example, plant lice and fungi, can also sometimes be acceptors. With sucrose are supplied by the symplast most intensively, substances by the symplast.
We have to, all right.
Supplied with organic substances by the symplast. Store apoplast or.
Right. So let me tell you one thing. Any plant which is having some fruits or root storage organ, basically what is happening here, phloem is being transported from its source to some organ where it is being unloaded, okay, where it is being taken off in some finished fruit product form or storage product form. In this case, this is a tomato plant. So what is happening in case of a tomato plant? The leaf is manufacturing the food, and this food is being sent to the tomatoes, the fruits, and the fruits are taking this sugar and converting it either into starch or it is almost somewhat converted into other carbohydrates. Okay. So fruit becomes a sink, the formation organ becomes a source. This is happening in favorable conditions. Okay. Phloem is an organ, is a transport system which transports the sugars from source to sink. In favorable conditions, this can happen from leaf to root or leaf to fruit. But in unfavorable conditions, for example, winters are there. Now the leaves have all fallen off from the tree. Even if the leaf is not falling, there is another problem. There is no sunlight. There is no sunlight for many days. Now what will happen? How will the plant manufacture food? It cannot manufacture food. So now what will happen is the root which has finished stored product, starch, it will convert the starch back into sucrose, and sucrose will be transported from root to leaf or root to stem. This is one type of unfavorable condition. Suppose I am cutting a tree 1 meter above the ground. Actually it is cut about 1 to 2 meters above the ground. Now what will happen? You have left some part of the tree stem above the ground. Now this is going to create whenever the trees have to be cut, this is the current very good practice today, that you do not cut the whole tree up till the root. You only cut 1 to 2 meters above the soil. Okay. Now what will happen? Every tree, every plant has some dormant buds, dormant meristem, apical meristem parts which are stored as buds. These are in dormant condition. When the tree top part has been removed or it falls by itself, suppose the tree is breaking by the weight of the snow or any other problem, now what will happen? The buds will now suddenly become active. Now the stored food in the roots will be supplied to the supplied to the buds. The buds will take this food, they will multiply, cell division will happen, and new branches, new leaves, they will form. New stem will be formed in some time. So phloem is a part which transports the food bidirectionally from root to stem or from leaf to the root. This is the bidirectional transport. Compare it to xylem transport, it is only unidirectional. Okay. So this was the unidirectional transport, bidirectional transport. So let's write a few points here. According to the pressure flow hypothesis, according to the pressure flow hypothesis, there is a source and a sink source is the part where food is manufactured. Food is manufactured or is available freely. Where food is manufactured and it is available freely. It is not bound to anything. Okay. Sink is the part where it is consumed. Sink is the part where it is consumed. Sink is the part where food is consumed or stored. Consumed or stored in insoluble form. It should not be soluble. Normally, food is stored in the form of starch. Normally, stored as insoluble starch, because starch is an insoluble substance, right? Very slightly soluble, more or less insoluble. Insoluble starch. Now let's try to understand this. There are two seasons, one is favorable season, the other is unfavorable season. So what is going to happen in the favorable season? Let's see. Have you written this? Have you written this, completed this? Please do it quickly. As soon as you have done it, I will scroll to the next part. Okay. Look, now the details have to be written down. What is going to happen in the favorable season? Alright, this is done. Alright, so let's move on. Alright, so let's see this. Look, source is the part where the sugar starts its journey. It is produced. Normally it is the leaf. Okay. Sink is the part where the sugar is ending up where it is needed to be stored. Okay. So, it will travel from leaf to sink. Okay. And phloem, what happens is, phloem and xylem both are situated very close to each other. Phloem and xylem both are situated very close to each other because they are part of the vascular strand. Now, when the phloem is being transported from the leaf to the phloem tube, wherever it is, like companion cell, wherever they are located, there is another phenomenon that is happening. When the phloem is loaded with sugar, it becomes a hypertonic medium. It becomes a hypertonic medium. And in this hypertonic medium, what will happen? If this is situated close to the xylem, xylem is having a lot of water, you remember? Students, please focus. I want you to understand this concept. Xylem is situated very close to the phloem. Xylem represents hypotonic medium or dilute water. Phloem represents concentrated solution. Highly concentrated sucrose is being put into phloem by active transport basically. Now, please tell me, what do you expect that is going to happen? So, what is the cell sap? Cell sap is basically the dilute medium that is present within the cell. Okay. Now, please tell me what I am trying to ask you here. Shillong students, let's first focus on this here. What will happen when phloem, which represents hypertonic medium, is situated close to the xylem? What will happen? Can you please try to guess what is going to happen here? Yes? Okay, Shillong, you are asking me cell sap. Look, I have already told you that there is an organelle within the cell. It is called the vacuole, and in the vacuole, whatever material is filled, it is called the cell sap. Okay. Cytoplasm is the medium that is present within the cell, and cell sap is the fluid substance present inside the vacuole. Whatever is filled inside the vacuole, it is called cell sap. Okay. Now, let's come back here. Now, what will happen? The thing is, the phenomenon that is going to happen, please write. Let's finish this chapter quickly. When phloem is loaded with sucrose, at leaf, the medium inside the sieve tube becomes hypertonic, or you can say psi W or water potential decreases. Immediate decrease in water potential. Okay. Now, let's write the next point. Water potential decreases. The phloem is situated close to xylem. Phloem is situated adjacent to xylem, and xylem contains dilute solution. Because we have just studied transpiration of water, you will understand that water column, nearly pure water, almost pure water is present inside the xylem. So it contains dilute solution or hypotonic medium. Dilute solution or hypotonic medium. Hypotonic medium. Right? Now what will happen? Water will move from hypotonic to the hypertonic medium because they are situated very close together. Because they are situated very close together. Situated very close together. Water moves from xylem to phloem. Xylem to phloem. So now this becomes a solution containing sucrose. It becomes a sweet solution. Okay, it becomes a sweet sugary solution. Sweet sugary solution. All right. Now xylem to phloem. Now the sugar in phloem moves from source to the sink, at the sink the sugar is consumed or converted into other form that is insoluble. That is insoluble. So let's write this quickly. Favorable condition. Favorable condition or season. Favorable condition or season. What is happening? Sugar is manufactured in the leaf, transported, loading of sugar into sieve tubes. It is unloaded at the root cells. Root cells, basically root parenchyma cells. And here it is converted into, unloaded at root cells, sorry. Unloaded at root cells. Okay, and it is converted into starch. Okay, now sugar is soluble starch is insoluble. Some part of the sugar, it will be consumed by the root cells, but most of it will be converted into starch. So you can write here, most of the sugar unloaded at the root cells is converted into starch. And some part will be used as food material by the root cells, because root cells cannot manufacture their own food, into starch. This is about sugar in favorable condition. Now in unfavorable condition, the situation will be reversed. Now please write unfavorable condition. Unfavorable condition. Unfavorable condition. During the unfavorable condition, what will happen is, starch, which is the insoluble form, it is first converted into sucrose. Now it is loaded, the site of or root storage organ. Normally the root only stores, but if it is stem, like any part of stem is also storing, root or sometimes stem also. Okay. Root or storage organ other than root. Storage organ other than root. Other than root. Okay. So the starch is the insoluble form. It is converted into sucrose and it is loaded into the other cells. Loaded into the phloem cells. Phloem, sieve tube. Okay. Conversion into phloem sieve tube. Now the phloem sieve tube transports, okay, transportation of sugar into the sink. This is the source now. Root is the source of sugar, or the the source of the sugar now in unfavorable condition, and now the sink will be normally the stem, okay, or buds, anything. Suppose the stem has been cut, then bud will be the sink. If the stem is there but the leaves have all fallen off, now the stem will take the reserve food. So this is happening in the unfavorable condition. This is the pressure flow hypothesis. Pressure flow hypothesis or mass flow hypothesis. Okay. So now that this has been done, this chapter is now finished. This chapter is now finished. transport in plants. This brings us to the end of the chapter, transport in plants. Okay. So now if you want a break, because now let's take a break, and after the break we will start with the next chapter called the mineral nutrition. Okay. So let's take a break, and after this break we will take the next chapter, that is the mineral nutrition. Okay. So don't take a long break, because you have already taken a long break, because I had seen so many people coming in the class. Okay. So it's fine with me, but just complete your notes. Okay. So don't take a long break. Okay, so please settle down. All right, please settle down quickly. Okay, so if we have finished this chapter, I have just sent you your notes, PPT on your Droppers group. Please take a look at that PPT. Yeah, it's regarding the transport in the plants. It represents a good summary of your chapter. So take a look at the summary also. I'll just recap through that summary. So, all right. So transport in vascular plants. Why does transport need to occur? Because material need to be transported. Okay. Transport within plants. You see that the plants are very tall, like these trees are very tall. And so one thing is xylem, the other thing is phloem. Passive transport, active transport, and diffusion. Okay. So all this part we have already done. There is nothing much to explain in this. The means of explaining osmosis by water potential. So it is based upon solute and pressure. Solute is due to salt or soluble component, and pressure is due to the cell wall. So water potential gives us an idea in which direction the movement of water will happen. Okay. So it is having this equation psi s and psi p. I have already explained to you. Psi p I have explained to you. All these components, please take a look at this. I have sent this on your phone also, so you can take a quick look how to understand all these things. And the physical pressure, everything, it is very clear. A lot of questions are also given in your module. You can take a look at them also, and please feel free to discuss with me if you have any doubts. Turgor pressure is just turgor pressure, root pressure experienced by a plant. The turgidity in the cells is the turgor pressure. Okay, turgid, flaccid. The one plant on the left is called flaccid, the one plant on the right is called turgid. So water and sugars move through long transport. Okay, so long distance transport requires xylem and phloem. Short distance transport I have already explained: simple diffusion, osmosis, plasmodesmata, and apoplast, symplast. These are the short distance transport methods which employ diffusion and osmosis. Long distance transport happens by bulk flow or mass flow. Bulk flow or mass flow hypothesis, pressure flow hypothesis by Munch, and the second thing called transpirational pull. Okay, all that represents bulk flow hypothesis. Bulk flow is a hypothesis in which movement through xylem and movement through phloem both represent bulk flow. Okay, and transpiration I have already explained. Okay, so all these things you already know. Okay, so pushing the xylem sap causes guttation also, and guttation can move fluids through only through few meters. Then xylem vessels are the elements which are involved in this, and tracheids are also involved in this. But in phloem, when we talk about phloem, the different phenomenon is happening. It is bidirectional, and in xylem it is unidirectional. So this is a very good diagram. Please take a look at the figures of the diagram, components of the diagram. You will be able to understand how this is happening. Okay. So all this is I have put this in this slide. You can take a look at that. Okay. So photosynthesis, transpiration, compromise, all these things are fairly obvious. Everybody knows it. Okay. So I'm just scrolling through this because all this has already been covered. So it is a summary. Okay. And stomatal opening and closing, it actually depends upon a potassium ion malate exchange theory. This theory I have not discussed here, but take a look at this because the theory will not be asked in great detail. Okay, so just you need to remember all these terms that I have put here. They will be in form of some question in your NEET examination. Okay. So these are the fairly, just go through this once. Okay, you will be able to understand what I am trying to tell you here. Okay. Sugar transport, that I was just discussing. an organ where sugar is being produced, sugar source, sugar sink. Okay, so all this is fairly obvious. Okay, and it happens by the pressure flow hypothesis or Munch's mass flow pressure flow hypothesis. Okay, so translocation through food happens through this. Okay, so this is a very good opportunity for you to revise all these topics by the help of this PPT. So I have all this I have given to you. Okay, so let's start the next chapter now, mineral nutrition. Everybody has received that PPT I just sent on your groups, right? So take a look. If anybody has not received it right now, let me know. I'll send it to you. Okay. So Churachandpur, Shillong and Senapati, all students must have received it. Tamenglong students, if you have not received, let me know in the chat box, or ask your coordinators to add me on your group if you have not received it. Okay. So Tamenglong student, if you have not received it, add me in your WhatsApp group. I'll send it to you. Okay. Continuing, mineral nutrition. Okay. So what do you mean by mineral nutrition? Minerals. What do you mean by minerals? Let's take a look. I have a PPT that I want you to understand by this. Okay. Okay. All right. Do you have received it? Okay. Thank you. I'm happy that you received it. All right. So you can take a revision. Everybody can revise by the help of that PPT. Okay. All right. So I'll give you more notes also, and the questions you will receive by Saturday evening. Okay. Questions for this chapter you will receive by Saturday evening. Okay. All right. So let's continue. Nutrients. Okay. Nutrients are basically required by plant. Okay, for its growth. Okay, and we have up till now identified 17 elements that are essential for the growth and full development of higher green plants according to the criteria laid down by two scientists whose name were Arnon and Stout. Okay, and how do you decide that a particular element or a nutrient has some importance or not? So there is a criteria that if the deficiency of that particular mineral element in the soil makes the plant unable to continue its life cycle, without that mineral element the plant should not be able to reproduce or produce flowers, and even if it is producing flower, there is no seed formation, something like this. So without that element, the whole life cycle of the plant must come to a full stop. That is a meaning of essential nutrient. For example, some plants contain gold also, some plants contain silver also, but is gold and silver essential for the plant? This is our purpose of having this discussion. So the deficiency of specific element in question can be prevented only by supplying that element only. We cannot supplement that mineral element by any other means. You cannot supplement gold by silver, or sodium by potassium, or potassium and sodium together by calcium. You cannot do that. Okay, that is the meaning of deficiency, and that element is involved directly in the nutrition of the plant. Okay, and it is quite different, because if you do not use that element, the whole metabolism of that cell becomes disturbed. These are the criteria. It should be involved in metabolism, and without that element, the plant must be unable to continue its life cycle. The third thing is that element is irreplaceable. You cannot substitute it by any other element. These are the three criteria by which we decide anything present in the soil solution, the mineral elements, the ions, whether they are essential for plants or not. Okay. So there are some elements for which it is doubtful whether they are really useful or not. So one such element is nickel, but we do not have nickel in our course. Okay. Okay. So let's see this element. There are some elements which have been discovered by scientists over the years. desaussurate carbon, hydrogen, oxygen, nitrogen, plant, potassium, sulfur, calcium, magnesium. Von Sachs and Knopp, they also made some criteria. Regarding these elements, the contribution of Von Sachs and Knopp is very important. Why? Because these people developed a technique called soil-less cultivation of plants. Do you people know about soil-less cultivation of plants? Can you really grow plants without using soil? Yes or no? Can you grow plants without soil? Yes or no? Do you know of that or not? And if you can grow, how do you grow it? No idea? If it's okay if you do not know about this. Let me know. Okay, no idea, I guess. Soilless cultivation of plants can be done in water or air. There are two things, okay. Take some plants, put them in a water solution actually, and add the nutrients to the water. Now there is one more problem. Soil supplies two things to the plant. Soil gives support to the plant, so you must support the plant by putting it somewhere. It should not drown in the water, okay. So soil gives support to the plant, support anchorage, and it gives water, it gives minerals, alright. So all these things must be supplied. You must give support to the plant, put some supporting thing, some arm which clamps the plant firmly. So plant must stay upright, hold it by the help of a plastic stand or something. Then give water to the plant, minerals to the plant, and soil supplies one more thing, that is air. Soil also gives air to the plant. If you do not give air to the roots will die, the plant will die. So you must give support, you must give water, you must give nutrients, also you must give air. So how will you give air? You must bubble air continuously like fish aquarium. You must bubble air continuously into the roots. These air bubbles will provide air to the roots and the plant will grow normally. Okay. Now Von Sachs and Knopp and some other scientists, they identified a unique way of knowing which elements are essential for the plant. How did they do it? The thing is, they found out that if you are growing a plant in soil, soil contains so many mineral nutrients, but it is very difficult to decide which is essential or which is not essential. So what should we do regarding this? They found out a way. They said that we will take pure water and we will take many minerals, and if you want to study the effect of one particular mineral, suppose you want to study the effect of absence of calcium or deficiency of calcium on the plant. Now what we will do? The thing is, we will make a water solution in which calcium is totally absent. I will add all other minerals, I will not add any calcium salt. Now I can study the effect of calcium deficiency on the plant. Similarly, sodium, add every other element except sodium. In this way, successively absence of one element or other element has been determined. Okay. So this is the way how we do it. Okay. All these minerals, do not make this chart, I'll already give it to you, no problem. Okay. So essential plant nutrients, mineral nutrients, all these are here. Okay. Essential plant nutrients. Framework elements are carbon, hydrogen, oxygen. Mineral elements are basically macronutrients and micronutrients. And these we need to discuss. These we will discuss. How do we decide something is macronutrient or something is a micronutrient? So just take a look at this table and then I'll explain. Alright. Okay, so just read this. Carbon, hydrogen and oxygen. Carbon, hydrogen and oxygen, they are basically obtained from CO2, and hydrogen is obtained from water, and basically CHO make 95% mass of the plant. All the 5% remaining is by the mineral elements, right? Now we need to define one more very important thing, and that is called the dry mass of the plant. How do you determine the mass of the plant? So plant contain a lot of mass of water. So before we need to decide that which element is present in which percentage in the plant, we take a standard, and this standard is called the dry mass of the plant. Dry mass of the plant will be used to determine the percentage composition of a particular element in that plant's body. Alright. So first, just take a look at this and I'll let you know. Okay. Alright. Macronutrients are those nutrients which are present in a particular concentration, and these are in more quantity. Micronutrients are present in a lesser quantity. Lesser quantity. Alright. Now let's study them. Iron, manganese, zinc, boron, copper, chloride, molybdenum, nickel, okay, and cobalt, sodium. Okay. So all these are micronutrients. Okay. Now this is a relative percentage in which these elements are present in that plant. Okay. Just take a look. You don't need to write it down. Just take a look at this table, and you do not need to write it down. Okay. All right. You do not need to write it down. Okay. Just try to understand it. Okay. Then now there are four groups, and actually in NCERT it is not mentioned like this, but this is just a way of expressing. There are four groups. In first group, the elements are present as organic substances. The elements are present as organic substances. Okay. And in the second group, it is present as basically involved in biochemical reactions. Okay. They are present as biochemical constituents. In the third group, these elements are present as ions, and the fourth group elements are present not as free form but as inorganic chelates. Chelates are basically inorganic complex ions. Okay. Chelates are basically inorganic complex ions. So now this is also a table. Carbon is absorbed in different form. Phosphorus, boron absorbed in different form. Okay. Actually, boron is absorbed as H3BO3 H2BO3 minus or HBO3 minus. Okay. All right. Potassium absorbed in this form. All the ions that are represented here, they are basically absorbed in one form or the other. Okay. All right. So this is the basic summary. Let's write the notes for this chapter now. Mineral nutrition. Mineral nutrition. soil is a complex solution or a reservoir of various minerals this soil is a complex solution or reservoir of minerals which contains both essential and non-essential minerals for the plant, which contains both essential and non-essential minerals for the plant. Essential and non-essential minerals for the plant. Now we need to determine which elements out of these are absolutely essential for the health of the plant. Okay, so we need to find out which of these are absolutely essential. To be able to determine I'm closing this. Okay, so it is all right. I hope you have already written. Okay. Next line. To be able to determine which element is essential, and which is not, a deficiency of that element must be created, of that element or ion must be created to know its effect, to know the effect. Okay. So how you cannot eliminate a particular element from the soil solution. You cannot do that. So we have to employ some other method. So please write down. Please write. The deficiency, the element or ion cannot be eliminated from soil, Instead, soil-less cultivation of plant, soil-less cultivation of plants is done soilless cultivation of plants is done to determine it. Okay. So soilless cultivation of plants is basically done to determine it. Okay. And this soilless cultivation of plant is called hydroponics or aeroponics. Hydroponics, please put the subheading hydroponics. Hydroponics. It is the technique for growing plants in water by adding minerals and nutrients to it, by adding minerals and nutrients to it. Okay. So let's draw a diagram how this is being carried out. We take a water trough, water reservoir like this. And we put a plant into it. Okay. And what happens is the root system that is there, I'm actually not showing it with brown color, I should have shown it with brown color actually. The root system that is here, it requires constant supply of air. For adding the constant supply of air, we take a tube and pump air through it, air bubbles through it. Tube for bubbling air. Bubble air continuously. And this is the water solution. And the plant is attached by the help of a support in such a way that it does not fall into water. So it is given the help of a stand or something like this. So the plant is kept in upright position and water is air is constantly bubbled into this water solution. So this plant is growing like this, and this plant will grow faster when compared to the soil cultivated plant. Why? Because it is receiving all the nutrients, basically mineral nutrients, and it is receiving a very good supply of air. So hydroponics is very faster. In hydroponics, plants go very faster compared to the plants of soil. Can you name me some fruits and vegetables? Can you name some fruits and vegetables where the this technique is being used. Edible fruits and vegetables that you are eating that are grown by the help of hydroponics. Do you know any some fruits or vegetables? Okay. How many people know salad? Salad or lettuce leaves? Salad or lettuce leaves? Nobody knows it? Okay. Are you familiar with seedless cucumber or the green colored, dark green colored European cucumber? Are you aware of about that plant? Sorry, that those type of cucumbers? Yeah, that cucumber that is coming in the markets these days, that cucumber is grown with the help of hydroponics. There are large water pipelines, and that plant is put, and now it also air is being circulated. The water that is falling from the pipe, it is constantly being circulated, and the mineral nutrient solution concentration is being checked from time to time. So in that way we grow seedless plants, sorry, not seedless plants, the soilless plants. This is hydroponics. Yeah, seedless cucumber is available to us. Lettuce and tomato. Tomato, there is one special variety of tomato, the small, very small tomatoes which are growing on tomato vines. That is the type of tomato that is being used, but we can use for commercial tomato also. But the smaller tomatoes are cultivated by this method. The larger tomatoes are being cultivated in the field. I am telling you what is actually on the happening in these days, because I am well aware of that. That is, I am telling you like this. Okay. So let's do that. All right. Time is almost nearly over, but you people came late, so I want you to sit a bit longer. All right. Now, how to determine criteria for essentiality? Criteria for essentiality of a mineral. Okay, it's time because it's holiday here, you won't get cab. Okay, all right. So in the next class we will start from here. Okay, all right. So in the next class we will start, basically this chapter will be finished in the next class. So 2-3 things remain: micronutrients, macronutrients, symptoms of deficiency, and the last part is nitrogen metabolism. So all this will be completed in the next class. Okay. All right. So have a safe ride back home. Okay. Thank you, dear students. See you in the next class. Thank you.